अस्सलाम वालेकुम।
वालेकुम सर।
कैसे हो बेटा?
अल्हम्दुलिल्लाह।
कौन से कैंपस पे हो आप?
रावी सर।
अच्छा, दैट्स ग्रेट।
टीसीएस।
दैट्स ग्रेट। तो क्या कुछ पढ़ लिया है स्कूल में?
सर आपसे ही पढ़ रहे हैं।
हाँ, तो मैं इसलिए पूछ रहा हूँ ना कि आपको पता भी है ना क्या कुछ पढ़ लिया।
हमने बायोलॉजिकल मॉलिक्यूल्स, पॉलीसेकेराइड खत्म होने वाला हो गया, खत्म।
खत्म हो
सेलुलोसिस
गया?
पढ़ना है।
बिल्कुल ठीक है, सेलुलोज हम पढ़ चुके हैं, पढ़ेंगे अभी।
पढ़ना है अभी।
अच्छा ठीक है। अच्छा जी, इसमें अभी हमने बेटा पहला चैप्टर शुरू किया हुआ है। और इसमें देख लें कि ज़ाहिर है आपने पहले ये पढ़ा हुआ है। और उसमें कोई जो डिफिकल्टीज हैं वो भी आप डिस्कस कर सकते हैं मेरे साथ। ओके।
जी सर।
और उसमें आज हमने जो डिस्कस करना है वो है लास्ट लेसन में प्रेपरेशन ऑफ माइक्रोस्कोपिक स्लाइड, टेम्पररी माइक्रोस्कोपिक स्लाइड हमने डिस्कस किया था। एंड यूज ऑफ अ लाइट माइक्रोस्कोप टू व्यू अ स्पेसिमेन अंडर लो पावर एंड अंडर हाई पावर, जो अब हमने डिस्कस किया था। ओके। और टुडे वी विल कंटिन्यू विद दैट एंड we will discuss units of measurements of length. We often get questions on this topic. Generally, they are easier questions, not difficult questions. But as biology students are, bit, reluctant in making calculations. So even if there are simpler calculations, sometimes they fail to understand that appropriately and fail to answer them. So this is units of measurements of length. The standard unit for length is meter. This is known as an SI unit. Okay. And there are larger derived units of meter and there are smaller derived units of meter as well. We will not discuss the larger derived units like kilometers, etc. We'll discuss the smaller derived units that include, centimeter, which is represented with cm, millimeter, micrometer, and nanometer. Okay. So if 1 meter has to be converted into centimeters, we will multiply it with 100, as 1 meter has 100 divisions of 100 centimeters. So each centimeter is actually 100th part of a meter. Okay. If a centimeter has to be converted into a millimeter, we will multiply it with 10. So 1 centimeter is equal to 10 millimeter. And if a millimeter has to be converted into a micrometer, it has to be multiplied with 1000. So 1 millimeter is equal to 1000 micrometer. And 1 micrometer will be equal to 1000 nanometers. So if micrometer have to be converted into nanometers, then you will have to multiply it with 1000. So 1 micrometer is equal to 1000 nanometer. Okay. But on the other hand, if a centimeter has to be converted to meter, so then we will divide with 10, divide with 100. Okay. When we convert a larger unit into smaller unit, then we will multiply. And if smaller unit is converted into larger unit, like centimeter has to be converted into meter, then we will divide by 1000. Similarly, if millimeter has to be converted into centimeter, then we will divide with 10. If micrometer has to be converted into millimeter, then we will divide with 1000. And if nanometer has to be converted into micrometer, then we will divide with 1000. So let's see an example. So if, for example, we have to convert 28 millimeter into micrometer. So what we should do to convert millimeter into micrometer? We can follow a simple calculation method in which we already know that 1 meter is equal to 1000 millimeter, or we can write it 10 raised power 3 millimeter. We also know that 1 meter is equal to 1000 into 1000 micrometer, or we can write it 10 raised power 6 micrometer. You will have to memorize these values so that it makes easier for you to make calculations in the exam questions. So this two equations can be used to convert this millimeter into micrometer. As you know, the left hand side is meter. If this side is equal, it is same, then obviously right hand side should also be same. Right hand side should also be same. So it means 1000 millimeter will be equal to 1000 micrometer, or you can write it 10 raised power 3 millimeter is equal to 10 raised power 6 micrometer. So if we simplify it, so 1 millimeter, this can be calculated. This is simple mathematical rule. If we have to convert this 10 raised power 3 millimeter to 10 raised, to micrometer, we should divide it either by 10 raised power 3 here and 10 raised power 3 here. So this would cancel out this. So 1 millimeter will be equal to 10 raised power 3 micrometer. Or you can say 1 millimeter is equal to 1000 micrometer. So to convert 28 millimeter into micrometer, we should multiply 28 is multiplied with 1000, and that would give us 28,000 micrometer. Hope you have understood it.
Yes.
Right? So this is how we can have millimeters converted into micrometers, or micrometers may be converted into millimeter. We can convert larger units into smaller, or smaller into larger. We'll discuss some more questions. Let us see. So let's see. We have questions on conversion here. Okay. Uh, so we have this question. The dimensions of viruses are usually stated in nanometers. Okay. As this, there is a name of a virus. P. salinus is so big, it has been described as 1 micrometer long and 0.5 micrometer wide. Convert the width of P salinus to nanometer. So we have to, this is wide, so we have to convert 0.5 micrometer into nanometers. So to solve this question, either you should remember and you should know that 1 micrometer is equal to 1000 nanometers. Okay. So you can multiply this 0.5 with 1000 and you will get answer in nanometers. Or you should know that 1 meter is equal to 10 raised power 6 micrometers and 1 meter is equal to 10 raised power 9 nanometer. Okay. So as this left hand side is equal, so should be the right hand side. And this would give us 10 raised power 6 micrometer is equal to 10 raised power 9 nanometer. So if we have to find 1 micrometer, what you should do is divide it with 10 raised power 6 and divide it with 10 raised power 6 as well. This would cancel out it, and this would cancel out 6 and 3 will be left behind. So 1 micrometer is equal to 10 raised power 3 nanometer. Or if it is 0.5 to be converted into, this is 0.5 micrometer to be converted into nanometer. So we will multiply it with 0.5 here, and we should multiply it with 0.5 here. So we will get 10 raised power 3 multiplied by 0.5. So whatever answer we get, that would be what?
500.
500. So this should be 500 nanometers. So that is how we can easily convert one unit into the other unit. For example, if we are given width of an object is 500 nanometer. To convert it into micrometer. What we should do now? So we are converting from nanometer to micrometer. So we should divide this with 1000. And when we are converting micrometer to nanometer, we should multiply it with 1000. Is it okay? So 500 divided by 1000, that would give us again obviously 0.5 micrometer. So this is what we have done in this case. When micrometer is converted into nanometer, this would give us 500 nanometer. Unit is given already there, right? So now let's see the next topic. Magnification. Magnification can be defined as the number of times an image is larger than the actual size of an object. The number of times an image is larger than the actual size of an object. Okay, so this is the measured size, or maybe the size of an image, and it is divided by an actual size of an object. So this would give us magnification, or we can write it as M is equal to I over A. So M stands for magnification, I stands for an image size, and A stands for an actual. We will use this formula to make calculations of magnification. So let's see. Before we discuss exam questions, so let's see the magnification of a light microscope. Magnification of a light microscope. So a light microscope, you know, it has an eyepiece, and it has objective lenses of different powers that are attached with this part. It has a stage here, and you know, it has an electric lamp here, and this is, you know, base and the body that hold these structures together. So this is an eyepiece. Eyepiece may have different powers printed on it. Generally, an eyepiece that we use in the school lab is an X20. And then we have these objective lenses. The power of objective lenses, this is an objective lenses. We can have objective lenses of different powers. The smallest one is red in code, and it is X4. Objective lenses have different powers. The smallest one is X4. Then the next is X10. Further higher is X40. And then another we have is an X100. These two, X4 and X100, are not in our syllabus. This is used for scanning and starting setting a microscope to view a specimen. In exams, we will not be provided this objective lens with our microscope. We will be provided two of these objective lenses, one with an X10 and other with an X40. However, to calculate the magnification of, you know, what will be the total magnification of this light microscope can be obtained by multiplying the power of an objective lens with power of eyepiece. If eyepiece power is 20, so we will multiply each with 20 to get the magnification of a light microscope. So magnification will be at this lower power, you know, this is X4, we will have 80x. Okay, and at this, if 10 objective lens is used, then we will have 200x. And if we use this 40, then we will have 800x. And as we do not use this last one, but if it is used, then we will have 2000x. So this is the maximum magnification. This is the maximum magnification, max magnification of a light microscope. Okay. Whereas the magnification of a light electron microscope is 250,000 times. magnification of an electron microscope, EM, that is 250,000 x. We can write x after this number, or we can write this x before this number, like x 250,000. So this x is actually for number of times. Okay. So by changing the power of the, by changing the power of lenses, we can change magnification. Okay. And magnification is limited by the resolution. If a microscope has less resolution, then obviously the magnification cannot be increased more than certain maximum. For example, if a light microscope has, it has x 2000 magnification, so the resolution, we will discuss about the resolution, what does the resolution mean, and then we'll come back to this point. So if we increase the magnification of a light microscope above this 2000 times, then the image seen will be a blurred image, so we will not be able to see. So this magnification is restricted or limited by the resolution of a microscope. So let's see, what does resolution mean? So this is resolution. I'm sure you know this is this word actually comes from resolving something, or this comes from separating something. So resolution in biology, that means ability to distinguish two objects separate from each other. Okay. Light microscope has a resolution of 200 nanometer, whereas a scanning electron microscope, this SEM stands for scanning electron microscope, has a resolution of, this is the resolution of 6 nanometer. Whereas TEM, there are two types of electron microscope. One is known as an SEM, which is scanning electron microscope, and other is known as TEM, transmission electron microscope. So transmission electron microscope has highest resolution. Transmission electron microscope has highest resolution, and it is 0.5 nanometer. So what does these resolutions of 200 nanometer mean, and resolution of 6 nanometer mean, and resolution of 0.5 nanometer mean? So resolution of 200 nanometer means resolution of 200 nanometer means objects lying at a distance less than 200 nanometer. If this distance is less than 200 nanometer cannot be seen as two distinct objects. So if the distance is 200 nanometer or more than 200 nanometer, then only these objects can be distinguished as two separate objects. Okay, so if two objects have a distance less than 200 nanometer, less than 200 nanometer distance, then these two objects will not be separated as two different or distinct objects. Instead, we will be able to see this single object. This will be a single object seen under light microscope. So this is what does 200 nanometer mean. So a minimum 200 nanometer resolution means a minimum distance between the two objects to be distinguished as two separate objects. Hope you have understood it. Now, what does resolution of 0.5 nanometer mean? This is the resolution of transmission electron microscope, or TEM. So it means that the objects that are 0.5 nanometer apart, these two objects are at a distance of 0.5 nanometer apart, can be or are distinguished as two separated or distinguished as two objects under TEM. So TEM has higher resolution. This is higher resolution. This is higher ability to distinguish the two objects. So this is the higher resolution. And this is lower resolution. So what factors now affect the resolution of a microscope? The resolution of a microscope wavelength of light or radiations used to illuminate an object determines the resolution of a microscope. Okay. Power of lenses does not distinguish or does not determine the resolution. Okay. If we change the power of a microscope or the power of objective lenses, the resolution will not change. The resolution will change if we change the radiations which are being used to illuminate an object. For example, if we look at a white light, which if is passed through a prism that is separated into 7 different colors, It is violet, indigo, blue, green, yellow, orange, and red. Violet has a wavelength of 400 nanometer, and red has a wavelength of 700 nanometer. Okay, so this is the shortest visible wavelength of light, and this is the longest wavelength of light visible to human eyes. So if we use a light microscope and the specimen is illuminated, or the violet lamp is lit, or violet filter is used, then the resolution will be half of it. The resolution will be 400 divided by 2, that would be 200 nanometer. But if the specimen is lit by the red light, then the resolution will be 700 divided by 2, that would give us 350 nanometer. 350 nanometer. Okay, so if the microscope is lit by a red lamp or red filter is used, then the resolution will be higher. Sorry, the resolution will be lower. This may be sometimes confusing to the students, that this is a 350 and under the red light, and it is 200 under the violet, in the violet light. So which one is a higher resolution? So smaller is this value, higher will be the resolution. This is higher resolution, and this is lower resolution. So tell me, if a violet lamp is replaced with a red lamp under a light microscope, what will happen to, will the resolution decrease or increase? So you see, as I said, under the violet lamp,
Decrease होगी sir.
yes, it will decrease. So when the violet lamp is used, the resolution will be higher, and when we switch it from the violet filter or the violet lamp to the red light or the red lamp, what will happen? The resolution, the number will increase. Here in the violet lamp or violet light, the resolution will be 200 nanometer, and when it is replaced with red, as we already discussed, it will be 350 nanometer. So resolution will decrease. Okay. However, as we increase the magnification, if magnification of a microscope is increased, the image will blur. Why it will blur? It will blur due to the magnification being limited by the resolution. Okay, if lower will be the resolution, we will be able to magnify an image to a lesser extent, and if there is a higher resolution, then we will be able to magnify an object to a larger extent. Okay, so if we increase the magnification of a microscope, then the resolution of the image will decrease. The resolution of the microscope will obviously remain the same. Means that would be determined by what kind of the radiations or what wavelength of radiations is being used to illuminate an object, but image will become blurred. So that's about magnification and resolution. So let's see some question on magnification and resolution. Then we will continue with the use of an EPG in the next class. So let's see. This is calculate magnifications of images and actual size of specimens from drawings, photographs, and electron micrographs, scanning and transmission. So there are actually three types of images. Number one, photo micrographs. These are images seen under light microscope. Okay. And there are images that are seen under scanning electron microscope, and these are known as, scanning electron micrographs. And other are transmission electron micrographs. So this is TEM. Okay. So what are the differences in these three different kinds of images that we get? So you see, these seen under light microscope would obviously have lesser magnification up to 2000, and they would have lesser details seen under them under a light microscope. Scanning electron microscope has surface details, and transmission electron micrograph will have internal details. So this would have highest resolution. So internal details will be seen more clearly, and more details will be seen under a transmission electron micrograph, in a transmission electron micrograph as compared to the other two. So these are the three different types of images. If we look at this cell, so we see the internal details. So this is actually an electron transmission electron micrograph. Okay, as there are internal details seen as well. So that this is, transmission electron micrograph. Okay, so the actual diameter of nucleus between X and Y is, this is an actual diameter. This is 9 micrometer. Calculate the magnification of the plant cell shown in figure 2.2. Write down the formula for magnification and use it to make your calculation, show your working. So formula is simple. You should write, either you can write abbreviations, MI over A, that's fine. Or you should write in words, magnification, so that doesn't matter. But you should write either of them. There is no need to write both of them. So this is image size over actual size. Okay. And then how you should make a calculation. This is for 3 marks. What you should do is, you should take ruler and measure this length in millimeter. So this is step number 1. Once you have taken this measurement, then the second step is convert to micrometer. Okay.
Sir.
Yes.
ये इम्पोर्टेंट है कि माइक्रोमीटर में करें, हमारी मर्जी, हम दोनों को कोई भी उसमें करते हैं, यूनिट में करते हैं, पर होने दोनों सेम चाहिए.
They should be both the same. Okay, you can convert this micrometer into millimeter. If you make measurements in millimeter, you can convert this micrometer into millimeter, so that makes no difference if magnification has to be calculated. But generally the calculations in the unit which is used in microscopy is micrometer. Okay, And it is also easier to convert a larger unit into smaller unit. So millimeter will be converted into millimeter is multiplied with 1000 to convert it into micrometer. So this is an easier to make a calculation. On the other hand, if these micrometer have to be converted into millimeter, we will have to divide this 9 by 1000. So this would convert it into millimeter. So you can do either of them. Okay. So let's see if we measure it. For example, if we measure it with a ruler and we assume we cannot measure it right now because this is not an actual size on a paper. So we assume that this is maybe 25 mm, for example. We have just assumed it. We used a ruler and we measured this length. And this is, for example, 25 millimeter. So this is an image size. This is image size. Okay, we will convert it into, this is step number 1. And number 2 is we will convert it into micrometer. So number 2 will be 25 and 2000. So this would give us 25,000. And this is an image size. Then we have an actual size, which is 9 micrometers. So magnification will be 25,000 divided by 9. And this magnification will be, this is X already given here. So you will make a calculation and you should give this value here. Okay.
Okay, sir.
And luckily, this measurement made has been, the 25 millimeter. So if this measurement is exactly 25 millimeter, this is how we will make calculation. And you should accept +1 and -1 millimeter in measurements. When you are making measurements, you can make, an error of +1 or -1. So if you take 24 millimeter or 25 millimeter or even 26 millimeter, that would be acceptable. Okay. So this gives us 25,000 divided by 9, and whatever answer you get, you can write that here. So this would give us 3 marks. 1 mark for, this writing this formula, 1 mark for writing this, the step, calculation step, and 1 mark for writing this correct answer. Okay. Now we have another question. The electron micrograph shows a type of a virus at magnification of 30,000. Okay, this magnification is given. Magnification is given. And what is the length of the virus? So actual length has to be, is to be calculated. And the formula we know is M is equal to I over A. If actual has to be calculated, then it should be I over M. So how we will get the I value? M is, M is 30,000. And what will be this I value? I value should be taken from here. Okay. Measure this length with the help of a millimeter ruler. Use a millimeter ruler. Make this measurement. Okay. So as we cannot measure an exact length from here, but then look at the other step. All these answers are nanometer. So we will have to convert this, millimeter measurement with the ruler into nanometers. Okay. So assume if it is maybe 22 millimeter, for example. So how we will make calculation if it is 22 millimeter, for example? So we know 1 millimeter is equal to 1000
से multiply करवाएंगे इसे.
We will multiply with it with what?
1000.
No, we will multiply it with 1000 to convert it into micrometer. We have to convert it into nanometer. These answers are in nanometer, not in, you know,
Same. Okay.
micrometer. So we should convert them into nanometer. And to convert this, and 1 micrometer, you know, is equal to 1000 nanometer. So consequently, 1 millimeter will be equal to 10 raised power 6 nanometer. Okay. So we will have to multiply this with 22 into 1000 and 1000. And we will have 22,000 and 1000. So this is, you know, in nanometer. Then divide it, you know, 22, maybe because they are also in, you know, 10 raised power of 10 raised power of 10. So instead of writing this value, we should write it 22 multiplied by 10 raised power 6 and then divided by 30,000. Let's see what answer do we get in this case. Do you have calculator?
Yes, sir.
What answer did you get?
733.
Sorry?
733.
733. Let me calculate it. Yes, it is 733 nanometer. Okay. So we can write it in scientific notation. So if we, you know, put a decimal point here, 7.33. So what we should do now? We should write, as we have
10
moved,
raised to power 2.
10 raised power
2.
2. Yes. So that should be nanometer. This should be an answer. So the actual value, obviously, image value that we will measure with the ruler, that would be different from what we have assumed here. So we will have a different answer here. Okay, but this is how we will calculate this answer. So that's all about magnification and resolution today. In the next class, we'll discuss use of an EPG, and we will discuss questions on the past paper questions on an EPG. So that's it for now. See you in the next class.
Allah Hafiz.
Allah Hafiz.